Good afternoon, and welcome back to where we left in the last class. And we were discussing about real-time PCR, right? Real-time PCR. We have to discuss the objective of real-time PCR is to, or the principle, or the overall principle is being able to estimate the product, amount of product formed in real time. Real time means in this case it is after each round of PCR reaction, PCR cycle. So if you want to do that, and how can we estimate the product formed is the question. The product that is being formed here is double-stranded DNA, right? As a typical PCR, we will have double-stranded DNA formation. How can we detect it? The last class we have discussed about two different strategies, and one of it is cyber green, which is much more safe, inexpensive, but maybe we have to compromise a little bit on the sensitivity. The other one is using Attackman Probe. That is what we are going to revise now. So, or else in the cyber green, it is simple. It is a fluorescent molecule. It will fluoresce only if it is bound to double-stranded DNA. Otherwise, it does not, no fluorescence if it is not bound to. So after each round of PCR, two products will give four products will give eight, and so on. You will have this twofold increase in the products. That means the fluorescence should also increase. By quantifying the fluorescence, we can estimate how much amount of product is forming, right? So that is real-time PCR. It can be used for many, many purposes. And one of the important purposes that people use is to quantify the template. The template, when I say template, it could be any of the things, something like it could be mRNA, it could be some other RNA molecule. And if it is RNA molecule, we would have gone by reverse transcription reaction first, and then real-time PCR. You can also do it for other pathogens as well, using small DNA and so on. So the Attackman Attackman probe is something special in that it is a kind of something similar. It's an oligonucleotide, single-stranded, and on one side we have the reporter, the dye that can fluoresce, and the other one on the other side end you have a quencher. If these two are present in close proximity, then there is no fluorescence, because the effect of the quencher will not allow this reporter to fluoresce. So the typical setup will have a forward primer, a reverse primer, and a Attackman probe like this. And when DNA polymerase is added, PCR polymerization would occur in this direction on the lower strand, and in this way on the top strand. So we have to choose a particular type of the DNA polymerase, which has 5' to 3' exonuclease activity. So when it encounters a junction, when it is within a junction like this, where the probe has come, the DNA polymerase is polymerizing, and slowly it has come till here, and now it is obstructed by this probe that is bound here. So the DNA polymerase that is present, the DNA polymerase using its exonuclease activity will break the probe into pieces, something like in the Nick translation. And then the strand is synthesized continuously. So as a result, what is happening is the reporter is physically distant from the quencher, because of which now the reporter dye will fluoresce. So every time we see fluorescence, which would mean a free R at the reporter, which would, if it is free, it would mean that it is degraded. That means it means that that product is formed. This is the product, right? Product is formed like this. That is the meaning of it. So after each round of PCR, the machine will check how much of fluorescence is present in each of the wells, and that is how the amount of fluorescence is estimated after each round, and it draws a graph something like this. As I was discussing in the last class, every machine has a limit, detection limit, and only within this region, between somewhere to here, the absorbance is proportional to the quantity. So if there is more template, then the formation of the detection of it would be much faster, and then we will see an increase in the concentration of the product. Here we are detecting fluorescence and calling it as concentration of the product. And beyond this, it could have been more, but the machine cannot, probably there will be a machine limit for that as well, where it is not proportional. So if a product is detected early, like in this case, that means the templates must have been higher. So this point is called as something like CT value. I have circulated some YouTube videos on RT-PCR, real-time PCR. I hope you have gone through them. If not, please go through it again. So in this case, A, B, C are three different samples. Each of them must be having about the same. It is the same sequence that we are trying to figure out. We are trying to quantify how much of the template. So if you template, how much of template is present, and if it is giving lower CT values, that means the template is high in concentration. Because of this COVID and the pandemic, I think now many people know about what is a. Just give me a minute, please. Sorry for the interruption. Yes, I was talking about the product, and if the CT value is high, CT value is low, which would mean the template is high. And now, because of this pandemic, everybody knows about, everybody has heard the word RT-PCR, right? And we, as biotechnologists or bioengineers, biology students, we should know how it is done. Typically, what they would have done is from the sample, they would have taken the sample, and from the sample, they would have isolated the RNA using a kit, total RNA. And from the total RNA, they would have used primers with Attackman Probe that are specific to the most solid, most conserved region of the COVID-19 RNA. And then they would have performed the RT-PCR. So if the values are very low, or the product is detected very early, then it means it is presence of COVID in it. The other kit, I think we will discuss this later too. It is about rapid test kit, right? I would want you to have a look at that, and next time in the next CIA, you will have a question based on COVID-19 diagnosis. Okay, it's a part of our syllabus too, and we have some advantages and disadvantages of RT-PCR method and the rapid test method. And I would want you to have a look at them, try to read the literature properly, and understand it well enough so that it will be useful for your CIA exam. The second one is the next one. What we are trying to do is find out. It's called as RNA in situ hybridization. RNA, we're trying to detect RNA. In situ is while within, it is still within the cell or so, in its location or site, you can think of it. And we are using the technique of hybridization. Hybridization is the base pairing like this, like we have done it in southern blotting, northern blotting, and so on. Just 1 second, please. Yeah, sorry. So here is a method where we have the cells, and the objective would be simple to find out if a certain RNA is expressed or not within a cell. If there is a gene, there is no guarantee that the gene is expressed, right? We want to find out which gene is expressed. Or if you have a tissue, in a tissue, some cells may be expressing the RNA, or we want to know which all cells express this particular cell, this particular mRNA. And in those situations, this is a good way of trying to do localizing, finding out which cells or which parts of the body actually express this gene. It's almost like northern blotting, except there are some variations. In this case, we will also see in the next slide, which will have a much more detailed one, but yeah, we will repeat it twice. So we first have to make a cDNA complementary to the mRNA. It is something like the probe, and that probe should be complementary to the mRNA that we are trying to seek. Okay. And then this is the probe should have a detection system. Somehow we should be able to detect. In the previous ones, we have used radioactivity. And at that time, I also said there are other ways, non-radioactive ways also possible. And here is one which we will discuss in the next slide. Assume that there are some molecules that we have added in the probe. So first thing, once we have the probe, we can take the sample and treat it with detergent, and then the cell membrane is opened up. So mostly this is used for, say, animal cells, because animal cells do not have cell walls. Fungal, bacterial, and plants have cell walls. So probably this is not so useful for other types, unless we break open we have a step including where the cell wall is broken. So using detergent, we can break open the cells partially. We don't want to completely break it. So we're just kind of making holes through which the probe might diffuse inside. And we have the molecules that I said I will describe in the next class, next slide, which is digioxygenin on the uridine labeled, and that will find its way. And if the mRNA, wherever in the cell, wherever it is present, it might base pair or hybridize like this. So once that happens, we will wash it to remove any unbound probe, and then we will send in the monoclonal antibodies against digioxygenin. And that digioxygenin, which is the antibody against digioxygenin, or I will call it as anti-DG, is conjugated to an enzyme like alkaline phosphatase. And now we add substrate for alkaline phosphatase. A colorless compound is given. I can't remember the exact name of it, and because of which is acted upon by alkaline phosphatase, and the phosphate is removed, and the compound which was colorless now becomes purple or dark blue, and that is detected as the color detection says that the antibody has gone. Alkaline phosphatase is present. Alkaline phosphatase is bound to the antigen, sorry, antibody. Antibody is in the cell because the binding, because it was bound to the mRNA, sorry, the probe that is there. Why is probe here? Probe is there because the target mRNA is there. And when we discuss it in theory, everything is straightforward and it's simple, it's logical. But when we actually do it in the lab, it is actually a difficult one. One way it needs to optimize each and every step, the concentrations, and also we'd have to gain a lot of experience to get the things right, the timing, and so on. Let's look at it the same thing in a differently, little more details added on it. So, or else I will first summarize this here. We make a probe, first one, and probe is with digioxygenin labeled uridine, which we will discuss again. We will open the cells, hybridization, then we have a step, washing step, then treating it with antibodies, and then the antibodies will produce the color compound if it is there. So how is the probe made is one thing we want to discuss. I just now used something, the word or the phrase, Nick translation a little while ago. With the RT-PCR, the same way Nick translation is also used here. They take a gene of the for which the mRNA we want to test, and then we make DNA cuts so that the cut is randomly, it makes some endonuclease or cuts like that. Then we are going to use DUTP. UTP is normally, or uracil is normally present in RNA, and here we are using it as DUTP, that is an unnatural nucleotide within DNA. UTP, if it is there, it is usually with presence of ribose. So here it is DNA, so we are taking DUTP, and then it is conjugated to a compound that is digioxygenin, and we are going to use it to bind the antibodies to it. Instead, people also use biotin. And we add all the other DATPs, DNTPs, that is CTP, DATP, and DGTP. And then we are adding these also. What we are trying to do is we are labeling this molecule, the top one. Then once it has formed, we can hydrolyze or denature these two strands and isolate the ones that have the label, which should be the minus strand or the antisense strand, because we are trying to detect the mRNA, right? So we have to ensure that it is to be complementary with the strand that we are trying to detect. But the cells are fixed on slides, and they will be permeabilized a little bit using detergents, then denaturation to occur, and then you can also hybridize. So as opposed to the previous one, where we are doing it for RNA in situ hybridization, here the technique is almost similar, except that we are trying to do here, we can try to find out mRNA. Okay, so this doesn't have to always be RNA. So once the denaturation is occurred, we will hybridize the probe, and then washing steps are involved. Then we would have added antibodies against anti-DG antibodies conjugated with some enzyme, something like alkaline phosphatase, and then we can detect. Then the compound will give a color which would indicate the location within the cell where this gene is located within the nucleus. Okay, so this is another way of how you want to locate the gene presence of a gene or a sequence within a nucleus. In the nucleus, it is with more and more knowledge that is coming up. What we know is the chromosomes have a certain way of organization. We can't go into all the details, or I am unaware of all the details, but a general idea is something like this. Towards the periphery, the chromosomes, each chromosomes may have certain regions where there are high amount of transcription. All those genes from different chromosomes tend to come to one location like this. That is something like it is like a transcription hub within the nucleus. The chromosomes that are present on the periphery or closest to the nuclear membrane are usually the heterochromatin. That means they're not as much transcribed or expressed. And if we want to know which part of a chromosome or which gene is located in and where is it located within the nucleus, we can use these kinds of techniques to find the location of those genes, or even the mRNA, as in this case. Then it is RNA in situ hybridization. Okay, so we are still actually trying to find about gene expression only. Okay, this is a little bit of deviation from the RNA in situ hybridization. So the other one is, this is about mRNA. So we have a gene, and the gene is expressed from here. What we say as +1 means that is the first base added in during transcription. And from there on, transcription would have occurred. And here somewhere there will be a termination. That is where transcription would have terminated. We know upon translation, it would have given a product. But if we want to know the regulation and the gene expression, we need to characterize the full mRNA. This region would be called as, this is the 5' of mRNA, and this is 5', 3'. You call it as 5' UTR, this region. That is 5' untranslated region, or sometimes it's called as leader region, and this region would be called as 3' untranslated region. How can I find out the +1? That is the 5' end of the mRNA. How can I figure that out? And here is an experiment called as primer extension assay. This word would be used anytime we have a primer and we allow extension of it. I think I would want to take this some other time because I forgot that I should have finished the sequencing first. Sequencing requires, the Sanger's method of sequencing requires its own separate lecture. So we will discuss at that time, okay? Because I didn't talk about it. We need to talk about sequencing and then come to primer extension. Because if we are trying to find out just the tip, we want to know how many bases is it from here to here, it could be easier. But if you also want to know the bases that are present from here to here, then we would have to combine Sanger's method and this primer extension assay. So we will skip this for the moment because I have to take maximum Gilbert method and then Sanger's method, and then it will require more introduction. I'll discuss this too in a later time. So here are some of the other things which is for screening. And as a matter of fact, we are supposed to still discuss about Cdna libraries. I think we have skipped the Cdna libraries. We have to do the gene libraries. So before going in here, I would want to talk about something so that you will probably get an idea about how a question might come and how you are supposed to answer in the CAA. I have given you a gene or genome, okay, and assume that it is a fungal genome and it is new. Excuse me. I'm sorry. So there is a fungal genome, and you are supposed to, or I will put it this way: we found a fungus, new mushroom, and we know that it is producing somehow by protein purification. We have taken it, we purified the proteins, and we know that it produces amylase enzyme. And how can I get the gene? How can I get the gene for this amylase? So a question can be framed such a way that you will be given a sorry you will be given a genome, and you will be told the story about this novel genome or novel mushroom is newly identified one actually has produces amylase, which is very resistant and has better properties than the other amylases. And so we want to isolate this amylase in great quantity. So your job is to identify this gene. How would you do that? That is the question. And we will go through some of the ways of trying to answer it. They are not ideal, but it should help you in thought processes and different considerations that you should have. Can anybody try to help me, and then we can all go through this together? I hope you understood the conditions anyway. We have to isolate the amylase gene from this particular genome. How can we do it? Anybody? We can go through two strategies. One of it is gene library, and the other one is cDNA library. Which one should we choose and why? Okay.
Cdna, sir.
Why cDNA library?
Sir, in very basic ways, we would say that it's mainly coded for the, it contains only the ones which are the essential coding regions rather than the whole genome library.
Yes, exons, right? Exons
Exons.
are the coding regions, and the introns are the intervening sequences. So we don't want, because majority of the eukaryotic genes are in have introns, the intervening sequences, and upon transcription, the first transcript will also have these exons, and in the mature transcript, all the exons will be joined together. That is splicing, right? And if we take the gene library, the genes, because of the introns, can be very large. The first thing is, if we construct a gene library, we don't know if we are going to get full length gene or not. Maybe it would have some restriction enzymes that we are using, and it might be cut, and then none of the clones would be able to produce a functional genome. That's one probability or possibility at least. Secondly, it all depends on what host are you going to express it in. Say if I'm going to express in like the typical workhorse of the lab, that is E. coli, then E. coli is a prokaryote, and fungal one is an eukaryote. That means the sequences required, or the genetic information or material required for the splicing to occur, there is no splicing machinery in E. coli. So we cannot directly use the gene library directly like this, even if it is cloned intact. So in that case, the best way is to use the cDNA library. And what is cDNA library? We would have taken the mRNA. Library means all the mRNAs that are produced. We will discuss again in the next classes the gene library and cDNA library together. So we will have a the mRNA, mature mRNA would be isolated, and then we will make cDNA, complementary strand DNA, complementary of cDNA, complementary of the mRNA using reverse transcription, first strand, second strand. We can also add several PCR, a PCR step to amplify. We will read 3 or 4 methods of how to do, and we will be able to judge or try to see which one is better in the given conditions and so on. So we would have cloned just the mRNA onto a vector, the cDNA, complementary of the mRNAs. So the start codon would have been here, and this would have been the stop codon in one continuous reading frame. So for that, E. coli will not have any trouble. Only trouble, if there is any, would be about the ribosome binding site. It is Shine-Dalgarno sequence that should be present for the transcription to happen. And when we clone, we should have cloned it in a vector that is with a promoter that is recognized in E. coli. Something, say, for example, lac promoter, PLAC. So when you use IPTG, IP propyl thiogalaktoside, isopropyl thiogalaktoside, when it is added, it will induce expression from the PLAC, and then because of which whatever is downstream would be expressed, and we will have the protein or the enzyme in this case. So the question is, the point is, when you're reading about in rDNA, you would be tempted to go after the technique alone. I don't think that is enough. Technique is important. You should have known the principle of it and so on. But what you are manipulating, the good idea about the matter that you're manipulating is what is important. We are a biotechnologist, or we are biologists. We are working with biological systems. And if we don't know if we don't have any idea about the fungus or the E. coli, the whole technique is actually a waste. We will never be able to use it appropriately for the intended purposes. So when you are deciding what to do, you should be able to recollect about the fundamental differences or considerations that we have to keep in mind, based on which you're going to employ the technique. If you remember, fungus is, as with every other domain, you can say, fungus has its own specialities. It has cell wall that is made up of chitin, right? And it has something else, extracellular digestion. That means the enzymes are secreted outwards, and the enzymes would digest the food, and then the molecules are absorbed in, and they are transported to different parts of the outgrowth, and that is how the fungus works. How can I detect amylase? First of all, how can I detect? You have done some experiments. The biotechnology students must have done some experiments about it. How do we screen or isolation of amylase secreting fungi? Okay, this is a common topic at one point that used to be as a project one. How do we do that? Anybody, if you have some knowledge on it, downstream processing, you might have done some of those, or industrial biotechnology, you might have. So anyone?
So we grow the fungi in a starch medium so that those which could produce amylase in an extracellular way would be able to degrade the starch and utilize the nutrients. And when
That's right.
we do the iodine test to confirm the same.
That's right. We would have taken media with starch. And then what would happen? We would have spread plate or made some spots, say. And I will just draw the plate as like this. And we have starch everywhere. And assume there are 3 or 4 places where the fungi has grown. Okay. And how would we identify which one? We would have used, uh, we would have added iodine all over the plate, and iodine would label, would stick to the starch, and has the whichever has a clear zone around it, something like this, right? And that zone, that indicates that that particular species of fungi is likely producing amylase. And that is why is it producing amylase? This, in this region, the starch is, there is no iodine here. There is no iodine because there is no starch. There is no starch because amylase has degraded it, or yeah, say degraded, right? And then we now know that this is the fungi or fungus that is producing amylase. Okay. Now the objective is not we are going to go through rDNA, and I want you to produce gene, identify the gene for the amylase, so that we can probably eventually continue to make one whole of this and then overexpress just the amylase and then purify, and you have the downstream processing things, right? So in every part of it, you can see that there is the screening. This would be the screening ones. Screening is trying to figure out which organisms are producing the molecule of interest or enzyme or protein of interest. And the second part is the, we would can call it as the actual essence, that is the rDNA. We are now engineering. Here we have devised a method to screen. No manipulations, we just have a method to screen. And here we are having engineering ways of purifying. No manipulations, we are just purifying everything else, right? Here too, we are not manipulating anything of the organism. We are doing everything to the outside, that is providing media, providing growth conditions, iodine, and all those things. Similarly, here too, we would have changed the pH or used different compounds for precipitation and all those things. This is the center. If there is anything about bio, if you are manipulating the genetic information, and that is where rDNA forms, right? What was naturally occurring wild type, we have manipulated it, and we are trying to get the best way possible to increase the production and productivity. So if I gave you a fungi and or say 10 different fungi and told you to find me the gene, isolate the gene that is producing for the amylase, and all I say I would have given you is 1 out of 10 fungal samples that were given to you are producing the amylase. How would you isolate it? So in that case, we would have first gone through the screening, then the engineering part. In the bioengineering or the genetic engineering here, because I call it bio here because it's about the information. We are manipulating the genetic information. So I just said that. But otherwise, it is genetic engineering or recombinant DNA technology, where we should. It's not straightforward. We don't have any sequence information yet. There is no nucleotide sequence information. There is no amylase sequence information. We just know that it is an amylase. And without knowing sequence, how can you get the gene is the question. So we have to go through the construction of libraries in that case, right? Of course, we can use a lot of techniques about bioinformation, right? In the sense, we know if we know about amylases from the other organisms, several other organisms, we could have seen what kind of protein folds are likely, what is the active site from them. We could get some information or indications from the other already existing knowledge. But that is not sufficient in this practically. So we don't know the sequence and we have to isolate the gene. That's a difficult task. In that case, we have to go with construction of gene libraries or cDNA libraries. And to construct the gene libraries and cDNA libraries, we have to keep several factors in our mind about the host, right? And if you have constructed the gene library and cDNA library, and then you have plated, assume if it is E. coli, and say, and after that you have spread plate it. We will discuss about gene library and cDNA library in detail again. I want to have a discussion about how the progression of things happens. I've gone through papers, your CA1 papers. Many of them are good, but I think there is always scope for improvement about how we write through scheme. Okay. When the CAA happens in the second CAA, it will be on campus, and then you will be given enough time to write, of course. So I want you to have an idea about thought processes that you should employ when you are trying to solve a problem in this case. So what host did you choose? Where is the actual one that is given was fungi, and we are trying to use host E. coli. And if we have plated the gene library here, each one is having one clone. Each one is a clone. And how can we identify if there is any amylase producing gene? How can you say which of these colonies is actually having the amylase producing clone or the recombinant DNA? Because the problem is the amylase, if we want to establish the similar screening mechanisms like using iodine and so on, we can still use it. But what we have to consider is the enzyme should be secreted outwards, right? If it is secreted, then we will be able to detect it using the iodine starch ways. Otherwise, it usually becomes very difficult. So try to recollect whatever you have read before. There will be several genes, several aspects of it. And I will quickly have, say about 5 minutes. I'll give you one more example so that you can also think about it. I have isolated a novel bacterium which is resistant to ciprofloxacin. Okay, it's simple. I haven't identified a novel, or I gave you a set of bacterial species. Several species are given to you. The question is, I want you to, assuming there are some resistance, I want you to isolate the ciprofloxacin resistance gene. I might even ask you, I want the nucleotide sequence of the ciprofloxacin resistance gene. What is given to you a different bacterial species from sample from somewhere, say from sewage. And I gave you some sewage water, and I am asking you to get me nucleotide sequence of a specific gene. How would you go about it? Anybody? Yes, please help me out, and then we'll close it. You have 5 minutes to do that.
So first serially dilute the sample and then plate them in a ciprofloxacin containing medium.
Yes. So we'll have a plate, and then we are going to plate it on top. Yes, on containing ciprofloxacin. Then, so all will die except the ones that are ciprofloxacin resistance, right?
Yes,
Yes.
sir.
Okay. Then what would happen next? What should we do next?
Plate it to confirm it.
Okay, we will do patching or other things to confirmation. I'll assume that we have confirmed. We have isolated. We got one bacterial species which is resistant to ciprofloxacin. Okay, next.
And then we can sequence the whole genome if possible,
No
or we could
sequencing.
isolate the total RNA.
Yes, I should
We
have
could isolate
mentioned that
total
there are no sequencing facility. Okay. Okay, yes. Then now what should I do? Say, when we say sequencing facility is there or not. Okay, we will go with sequencing. Okay, you said we'll go by sequencing. Then you got the sequence of the whole genome, right? How would you know which of it is that of ciprofloxacin?
We could isolate the total mRNA and then go for a cDNA sequence kind of thing.
Um, are you going by? So why did we do sequencing here then?
Like this was an alternative, so the mRNA thing.
Okay, that's right. So one is directly, you complete this one, then we will go by it. The other way is to go by Cdna library, right?
Yes, sir.
What would you do with this sequence? We sequenced. How would we know which part of it is actually responsible for ciprofloxacin resistance?
So one way of doing it could be sequence the genome of those bacteria which are not growing in the ciprofloxacin medium and identify the ones which are not present there.
Okay,
Right.
A and B. This one has 10 genes, this one has 10 genes. There are, in both of them, 5 common genes are there. Okay, 5. These are common ones. So A has 5 different, which are not present in B, and B has 5 that are not present in A. How would you figure it out? That is how a common way actually.
So we
Yes.
can knock down each gene, or we can specifically do restriction fragmentation, and then we can
Yes.
clone the specific gene to the specific clones, and then again do with the ciprofloxacin medium. So the ones which actually have the ciprofloxacin resistant coding gene, those will grow and the others won't.
Yes, that is certainly one of the strategies. And one of the reasons why I'm taking this now is as we grow, as we learn more and more techniques, you should be able to build your own strategies. So other one you said is we will discuss about it, which is called or which is based on transpogenesis by insertional inactivation. Okay, we will discuss about that too at some point. So with this, we cannot directly know which gene is ciprofloxacin resistance gene. How can you do it with this Cdna library? With Cdna library, we would have made a host of plates. We would have gotten a library like this, which means that each of these colonies would have had a recombinant DNA with one type of gene. And if we were growing this plate in ciprofloxacin, then the ones um, that are sensitive to ciprofloxacin would die, and the ones that may have inherited got the ciprofloxacin resistance gene would survive, and the rest will die. In those conditions, we should ensure that the host that we are actually choosing to make the library should be ciprofloxacin sensitive, right? And then we can take this and go for sequencing, and we have tested the phenotype here, that the phenotype is ciprofloxacin resistance, and here we would get the nucleotide sequence. Then we have everything, we know what is happening. The same way we could also have used I will just wind up now, the gene library, right? Instead of constructing gene library, we can take gene library and go with the same, something similar like this. There will be some advantages to doing gene library, there will be some advantages and disadvantages are also there. Okay, if you have any questions I would like to take, but otherwise I will stop here. I think in the next class, we will go with Cdna libraries and gene libraries. I will start with gene libraries and then Cdna libraries, because it's important for these screening mechanisms and how we can make what would be your considerations if it is bacteria? What should you do if it is plant or fungi or animals? Okay, any questions?
So this is based on something you mentioned in the class today.
Yeah, tell me.
So I'm not sure if I've missed it, but you mentioned that the heterochromatid would usually be in the peripheral part of the nucleus, and the euchromatin would be at the center or the core of the nucleus.
Yes.
And therefore, it would act as a transcriptional hub. But when I thought about it, I thought the transcriptional machinery, it would be easy for them to access the euchromatin if it is in the periphery.
Why?
The accessibility.
Why would it be accessible? Why do you think so? Excuse me. Say.
Because so there are multiple
Sorry.
components and
Yeah.
factors which have to traverse through the heterochromatin to reach the euchromatin to identify the corresponding gene and the promoter region to bind with it. I thought if it is
So
in the periphery, it would be easy for it to search for the
periphery,
gene.
where is it coming from? What if all the genes that are required for transcription to be one place? And for transcription to occur, we need RNA polymerase, we need transcription factors, we need NTPs. And what if all these things are pouring into one place, and that all the genes that need to be transcribed are in one place? So the template can also be in one place. If templates were, I'm just trying to rationalize, I'm not sure if this is the correct answer or not, but I'm trying to just rationalize. If the transcription factors, if everything was on periphery, that is a vast space, and in that space for all of them to be in one place, transcription factor, RNA polymerase, and DNTPs in sufficient quantities, might not be that easy. Okay, I am thinking so. I'm assuming that it is how it is. But you can say, what logic do you think in a different way? Why would it be easier? That is, in the periphery is one question I have difficulty understanding. But otherwise, we will always look at the literature. Why
So
would it
again,
be easier?
the transcribed mRNA to reach the cytoplasm, again, that would be easier if it is in the periphery. It would get transcribed and then it will be easily transmitted through the nucleophores.
So moving
As for
out
the
of the
DNA,
mRNA, moving out, you're talking about?
yes, sir.
Yeah, I mean, mRNA moving out is not so much of a problem, because even if in the point is making the transcription happen is the big trouble. Why is it a trouble? It is something like individual ones that should become together like this. It is something like increasing or decreasing in entropy. This is more of orderness in a way, if you see. Okay, I mean, in thermodynamics ways, if you see, it is like that. And somehow the enzymes and all these components that are there should be able to push for the formation of mRNA or RNA in general. So they all need to be in higher concentration. And if all the transcription was happening in one place, then you would have higher concentration of all in one place. As you said, technically or from the logistics perspective, getting the mRNA out is the point, right? And there will be nuclear pores through which the mRNAs would have happened. And nuclear pores are not everywhere. The traveling will still happen. So I think to increase the way the reactions happen and for all of the, say for example, what is the function of nucleolus? We have come across it. I'll stop in a minute. Nucleolus, what is the function of nucleolus, or we know of nucleolus? Anybody? So these are something like the ribosome organizing centers. That is where all the rRNA genes are usually located into the nucleolus. It has its own, I think it is the RNA polymerase 3 that is active. It will keep on producing RNases. Sorry, it will keep on producing rRNAs. And the rRNAs and the ribosomes, the ribosomal proteins are exported in, and this is where the ribosomes are formed and they are exported out. So we need to also have space for different types of genes. In some places, rRNA are produced, in some places, tRNA are produced, and in some places, mRNA are produced. Transcription has to happen, and not all of them can be diffused. And I think it is logical for them because eukaryotes have 3 different RNA polymerases, and they cannot all be in the same place working together because of course their recognition sequences are different, and the type of genes they transcribe are also different. And I think it would make more sense for them to be in isolated areas. What is common would be that they would have required NTPs. That is common. Transcription factors are a little bit different in between these 3 different RNA polymerases. RNA polymerase is different, and the type of template, that is the the promoter regions, are also different. So that is what I think. Okay, but we can check on it. You can also look it up and let me know if you find something more. The general one, of course, when we go into specific details, there will be more reasons. The question here, I like the question, is that why should it be like this? Why should it be that in the core? Why should it not be in the periphery? That is a nice question. Yes. Okay.
Sure, thank you. So also with
Yes.
respect to the
Yes.
DNTP being a common participant in any other processes involved in the nucleus, that should
NTPs.
also come into the nucleus.
Only
The
NTP, right? Yes.
Yes.
NTP.
Wouldn't that be synthesized in the cytoplasm and then be transported into the
Yes,
nucleus?
NTPs are produced in the cytoplasm and transported into the nucleus. Yes.
So would there be a concentration gradient in which the NTPs are?
There is a reason why the cells should be very small, right? Cells should be small. Why are they not giant cells? There are some, but they are specialized. But otherwise, majority of the cells are small. It is to maintain volume, diffusion rates, and many other physical forces do act upon it. So when it is the NTPs, you're saying the gradient is there. NTPs and DNTPs. NTPs are 10 times more than that of the concentration of DNTPs in the cell. Okay, so there is always a constant supply of NTPs. DNTPs are required only during S phase or prior to that, right? In eukaryotes, that would what be the point. NTPs are something that are continuously produced, and there is a turnover of the RR and RNA all the time. Okay, so I don't think there will be gradient if you are going to have more number of transport for the RNTPs. RNTP is correct to say. Then you will have enough of entry inside. As you go inside, okay, the volume is becoming lesser, right? If you count the surface area to volume ratio, this is too much surface area in a way. And from all different directions, if you have, you can maintain some kind of uniformity from here to here, because this volume, the volumes are different, and you have so many entry points. The space is less here, right? Even if from this pore there are two molecules supplied, from all the pores, if you have 2 to 2 molecules, then you have reached a concentration saturation in one sense. Do you agree, or you have a? We can think about it.
Yes, sir, this does make sense and it is possible.
Yes, that's right. Yes.
And one last doubt, sir.
Yes,
With
those
respect to
who
the
others you can leave if you want to. You can stay too. Yes, tell me.
with respect to the compartmentalization aspect of it, um,
Okay.
that is one of the reason why I said it would make sense if it is in the periphery, because it will be easy for specific genes to be transcribed in that way. If everything is going to be concentrated, then the RNA polymerase concentration would be higher, the activators concentration would be higher, and therefore all the genes should get transcribed. But that would not
Yes.
be a case.
I will try to counter argue. Say there are 6 genes. Okay. How many RNA polymerases do you want? This is 9, 10, 11 genes are there. How many RNA polymerases do you require? Pol 2, how many of them would you require?
That depends on whether it is going to happen simultaneously or.
Assume they are happening within 1 minute. So if they are spread around in different locations, okay, then you would need how many, say, for example, how many RNA polymerases are there? Say, I will give you 10 RNA polymerases, and I'm telling you per minute, one transcription happens. Okay. How many transcripts can be produced this way? You have to consider spatial movement of the RNA polymerase also. And I want you to probably compare it with, I'm cooking it up. These are not necessarily the answers, but I'm trying to think along your lines, and that is good discussion. If all of them were here, right? And what would happen if RNA polymerase is synthesizing, and then it has to come back and synthesize again, right? For this RNA polymerase to be again synthesized within the time when this RNA polymerase is already being synthesized is difficult. Yes, I gave you 10 RNA polymerases, and this one is synthesizing, and if something is one of it has finished somewhere, it can restart here. You are not having isolated pockets of RNA polymerase where this is the amount of concentration for each of them to be maintained to maintain appropriate amount of transcript formed from each hub. In a situation like this, right, any RNA polymerase that is free can act on any promoter that is around, and then they can transcribe. That means there is no wastage of time. You don't have to wait for something else. I think that way it will increase the efficiency with which transcription can happen, even with minimal amount of RNA polymerase. I think we can actually model this if you have something about it, and look up if there is any modeling ways by taking into account the speed of taking into account of the concentrations, taking into account the RNA polymerase, the speed of RNA polymerase, taking concentration of each of them. You have to consider the transcription factors, the genes, and all those things, and test which of these is good. When you give same conditions, and of course, we have to add also diffusion rates and other things. Which one would be most logical? Okay, and that is what I think.
Yes, sir. This is actually possible, and it makes better sense of having a better efficiency. But
Yes, probably. Yes,
but
go
my
ahead.
problem is, what if there is some gene, or like say there are 10 genes, and out of them only 6 needs to be transcribed and 4 should not be transcribed? What would happen in
Yes.
that case if the activator is present? Or would
Yes,
that be
I
resolved
mean,
only by the pressure, because the concentration of all other
it
factors are
is,
same?
that's right. Eukaryotic ones are much more complicated, right? You have epigenetics. You have very complex ways of how genes are regulated in eukaryotes. So in that sense, we cannot absolutely say which one is going to produce in what efficiency. But we can consider some factors. We should be able to consider the strength of the promoter and whether the activators are present or not. These are what, and the mediator complex, chromatin remodeling complex, multiple things are there. And this will decide with which affinity the RNA polymerase is going to recognize or bind here and escape from the promoter, what do you call that, promoter escape, and initiate the transcription. So this is a much more complex thing than what we are discussing here. I was trying to discuss only in terms of the physics ways. Okay, in biological ways there are more things. You have the RNA polymerase, the tail, which is phosphorylated, and other kinds of modifications, which will also determine whether the speed with which RNA polymerase moves ahead, and there are several processing enzymes to bind. It's more complex than that. Yes, I think I will not have a right away answer for it. But this is what will determine which gene is copied in which the rate at which that particular gene is transcribed. It all depends on the activators, repressors, and many other complex proteins that are bound here. Okay?
Yes, sir. Thank you so much,
Yes,
sir.
yes, no problem. You're welcome. Thank you for the discussion. Yeah,
Thank you, sir.
see you.
We'll look into it further.
Yeah, please look into it, and I'm interested if you can look at the, what do you call, the nuclear